Group 41, creating recombinant DNA. So the first step in creating recombinant DNA is to identify and isolate the gene of interest to be cloned. Secondly, we isolated the plasmid from a bacterial cell, cutting it with restriction enzymes. Then, we obtained a gene of interest from DNA, cutting the DNA using restriction enzymes to isolate the gene. Insertion of the gene of interest into the plasmid vector. Then, transformation occurred. This is when the plasmid was imported back into the host organism. Selection of a transformed host cell for us to study, and cloning of plasmid and expression of selected gene of interest to produce a protein. So we took, for example, a human insulin gene and inserted it into an E. coli plasmid vector. We took our human insulin gene from a genomic library. We took our plasmid DNA from an E. coli cell. These two fragments are going to give us our rDNA molecule using restriction enzymes and ligase. So the restriction enzymes cut at specific points, and the use of ligase and base pairing of sticky ends reanneals both the fragments, giving us our rDNA molecule. So the rDNA molecule is produced and is transformed back into the host cell. Cloning occurs, and also expression. Cloning is when multiple copies of the rDNA molecule are produced. Expression. Donor gene undergoes transcription and translation to produce